So I'm currently a postdoc at UC Berkeley, but I just finished my dissertation at UC Davis. And I'll be talking about some of my dissertation work, addressing maybe a type of intraspecific variation that we don't think about very often, which is polyploidy. So polyploidy is abundant throughout the tree of life, right? It's very common in some animal lineages, but it's very abundant in plant lineages. And we typically think about species as either being diploid or polyploid, but species can be mixed ploidy, where they contain multiple different ploidy levels. And here are a few maybe like better known examples of mixed ploidy species, but it's thought that at least 16% of plant species are mixed ploidy, which is much more than I think most people realize, and that's largely a huge underestimate. And this is surprising, perhaps, because we know that polyploidy can affect evolution, right? We know that if you have multiple copies of your genome, it can prevent the effects of a deleterious mutation from being expressed, or it could slow the fixation of a beneficial allele. Additionally, you could have changes in gene expression where proteins from one subgenome are interacting with genes from a different subgenome to create novel phenotypes, like changes in flower color, which has been shown in synthetic and natural Nicotiana polyploids. You could also have phenotypic changes like increases in cell size. That's been really well documented in looking at stomata size, where if you have more DNA, you kind of just have to make room. And so as a result, it's led me to think about these bigger questions about, when is polyploidy beneficial? And if it is beneficial, why do we see this type of intraspecific variation when one ploidy level hasn't fixed over the other or hasn't been lost? And I've been investigating these questions in Andropogon gerardi, commonly known as big blue stem, which is a North American prairie grass that's quite widespread. It's a bunch grass, so it grows in a clump, as opposed to a turf grass that spreads across your lawn, and it's quite tall. It can be as tall or taller than me. Here I'm showing the historical range of North American tallgrass prairies in the light green, which is the ecosystem in which big blue stem's dominant, although it's found pretty much everywhere east of the Rocky Mountains. The darker green is the historical, or is the current range of the tallgrass prairie ecosystem, which is only about 4% of the historical range. And that's largely due to land use change. So understanding adaptation in big blue stem is important for mapping the trajectory of this ecosystem in future climates. As a polyploid, it's composed of two ploidy levels, 6X or hexaploid here in blue, and a neoploid or 9X in red. And both are found sympatrically. They're found in the same environment. They're neighbors across the range. And they're both very equally abundant, but they have slightly different distributions, where the 9X are more common in regions with higher mean annual temperature and lower mean annual precipitation, so more like xeric, arid regions. And we don't really know why this distribution exists, although it's been well documented by Chrissy McAllister. Importantly, the 9X have frequent meiotic errors, and that really leads back to the hexaploids. So the hexaploids are an allopolyploid where they have 3 subgenomes. Here I've colored that in different shades of blue. And the 9X are formed by an unreduced and a reduced gamete coming together from the hexaploid to make the 9X. And as a result, you have 3 copies of every subgenome in the 9X, creating effectively triploidy, and this leads to a lot of meiotic errors. So here on the right, I'm showing a chromosome squash from in telophase, where the chromosomes are segregating away from each other, and in the middle you have a bunch of laggards, or like chromosomes that didn't have pairs, so they're not separating properly. And because of this, we know that the 9X have much lower seed set, and any offspring that they do make are aneuploids, and so their contributions to the gene pool are likely limited or 0. This has been well documented through greenhouse crosses and cytological experiments. And so as a result, following up on those studies, part of my research I won't be talking about today was looking at the origins of the 9X, and we found that the 9X are neopolyploids, where likely each 9X is its own origin. It had its own whole genome duplication event. And here I'm just showing a kinship matrix where you can see the black boxes on top are the columns with the 9X, and they're all unrelated to one another. They have a very carry a value of like pretty much 0 to one another. And so what we think is happening in this population is that a 9X arises, and then it can persist clonally through growth through an underground stem called a rhizome. And they could last, in theory, up to 100 years or more. We don't really have a definite age on how long they can live in this prairie. And so what is going on with this range? Why is this effectively sexually sterile or sterile cytotype? How does it have such a large range? And one of the potential reasons could be that this whole genome duplication event confers some sort of adaptation or beneficial trait to these more arid climates in the southwestern United States. And to investigate this potential ecological differentiation, I planted a common garden experiment using 14 populations from across this range. They're from very diverse prairie types, as well as both a precipitation and a temperature gradient. Here they're shown on a precipitation gradient. Importantly, you can't distinguish the two cytotypes by eye in the field, so we were randomly sampling for ploidy, and so we ended up with most of our populations being majority hexaploid, so the white populations, and 3 of them having good mixed ploidy sample sizes, so those are like our tester populations, and those are in black. And the populations were planted in Columbia, Missouri, where the yellow star is in the center of the range, or pretty central in this range. The populations, the genetic structure of the populations is like there's roughly like an east and west genetic group, but they don't have a lot of differentiation between them, like at most 5% divergence, and there's not a lot of population structure or isolation by distance. In the common garden, we had about 80 genotypes that were replicated 3 times each, and it was a huge effort to transplant this many perennial grasses into the garden, and I had a lot of help. And while we were out there, we phenotyped about 15 traits over 2 years. And to begin, I wanted to look at whether or not the populations exhibited local adaptation. And so our metric of fitness here is relative growth or change in above ground biomass between year 1 and year 2 of our measurement. And this is a common measure of fitness in bunch grasses. And we would expect if the populations are locally adapted, that populations that were transferred from farther away from the common garden site, from a farther environment, would do more poorly. And that's exactly what we see. And this holds whether you look at geographic difference or difference in climatic variables. And then we can ask, okay, so do we see large-scale ecological or ecotypic differences between our ploidy levels and our populations? And here we're looking at a principal component analysis on our 15 traits. And the points are colored by home population, and the 2 ellipses are 90% confidence ellipses for that east and west genetic group. You can probably see there's not many clusters or patterns. It's like, what are you looking at? It's kind of a blob. So there's no real morphotypes we were able to identify. And if we color the points by ploidy, it's the same situation, where really we don't have any like distinct overall morphologies separating the 2. And that's largely because phenotypic variation is very continuous across the species range. Here we're looking at plant height measured in each year, and on the left we have our most northern populations. They're quite small. They have thin leaves and very high tiller density, and on the other side of the range is maybe our population from wetter climates, and they're much taller and have thicker, wider leaves. There's a lot of variation. It's just very continuous. And so maybe it's more useful to look at each trait individually. And we can do that with a linear mixed model, where just very briefly, ploidy is a fixed effect, and we're controlling for neutral population structure by specifying covariance among genotypes with a kinship matrix. And out of our 15 traits, we found 2 were significantly associated with ploidy, one being our fitness measurement relative growth. So on average, the hexaploids grew 2 times larger, and the 9X grew 3 times larger, which is substantially more and suggests that in some environments, the 9X may have like a competitive growth advantage. Additionally, we found that stomatal traits varied between the cytotypes. And so briefly for maybe non-plant folks, stomata are the pores through which they're pores on the leaf surface in which plants breathe, so they're really important for regulating how much water is lost from the leaf and also how much CO2 can enter. And we see that maybe as expected, if you're familiar with ploidy and cell size, that the 9X have larger, less dense stomata. And I'm going to try and convince you that this has an ecological importance in this species. So we know that in at least a subspecies of big blue stem called sand blue stem, that as stomatal density increases, so does stomatal conductance. So we would expect that the 9X would have lower stomatal conductance. They would be releasing less water back into the environment. And if we model this trait, including environment in our model, here described by two environmental PCs, we see that PCE3, which is associated with precipitation, where drier populations have significantly larger and less dense stomata in our population, suggesting that, yes, this trait that the 9X have might explain why they are found in these arid climates compared to the more like mesic regions. And so to conclude, we see that these very fragmented populations exhibit local adaptation, and that whole genome duplication may provide adaptation to these xeric climates in the species, either through changes in physiology, through like stomatal traits, or by changes in relative growth, making the 9X more competitive. But I'd like you to imagine like what the implications are of having a like almost sexually sterile cytotype dominate populations in the Southwest, where it can potentially outcompete the other cytotype, the one that has the ability to sexually produce, holding the adaptive potential for the population. I think we should potentially start to consider, at least in this species, the role of polyploidy and whether it should be managed in the population. There's more tests that we need to be run, but it does beg the question if we should limit how many 9X might be in a population. And with that, I'd like to thank a large number of collaborators that helped collect and phenotype and sequence, and agencies that provided permits and funding. Yeah, we have a great reference genome. We are very, very lucky. The Joint Genome Institute assembled a hexaploid genome assembly for us, chromosome scale, all 3 subgenomes are resolved, and it's completely phased. We can map those to that assembly and call triploid genotypes. I am happy to talk to you about how we did the genotyping, because I don't have time to talk about that here, but we had to use some unique approaches. In terms of genome sizing, we use flow cytometry for the majority of samples, and then sequence-based approaches for a minority of samples. Yeah.
Usually you see, very cool, very interesting data, but usually you see higher stomatal density as you go to drier environments, and we're seeing the opposite, which is really interesting. Have you done like water use efficiency, stable isotopes, or anything like that?
No, I really wanted to maybe measure like leaf isotopes, but I couldn't. It was a little bit more expensive of a phenotype than we could afford at the end of the study when we thought we should look at it. So I think that's definitely a next step, and also maybe like growth chamber studies to look at stomatal conductance under different environments if we really think that that's important in the species. Yeah. Yeah, so that's what I was showing, I'm so sorry, in this slide, that this is these are hexaploid populations as well, or the circles. So we think because they have those larger stomata that the 9X having them is like consistent with the species as a whole having larger stomata in arid regions.
So hi, I'm a postdoc at the University of Bern, and today I'm going to talk about bridging 2 different theoretical fields or approaches, you could say. So on the one hand, we have ecological interactions typically described by, let's say, Lotka-Volterra equations or their higher order versions. On the other hand, we have genetic interactions, which is more the focus of the evolution community, those who study fitness landscapes and so on. But of course, the two sort of coexist and intersect in nature, and that can guide the evolution of communities. So I'm going to talk about, or try to pitch to you kind of a theoretical framework which can combine the both of them, and I'll show you what the consequences might be for resource competition evolutions. It's still preliminary results, but I hope you get interested in the idea. So to begin with, firstly, genetic interactions, in my view, are best described by fitness landscapes, which is a map from genotype to fitness. Of course, fitness, what it means depends on the context very much. But one specific aspect of fitness landscapes or object associated with fitness landscapes is fitness graph, which is a partial order, meaning that it is a fitness relationship between mutational neighbors. So here I'm showing you a two locus and biallelic system, right? And where in this fitness graph, so each of the node is basically a single genotype, and then you have these edges connecting mutational neighbors. So you can go to a neighbor by mutating, provided the arrow is pointing in the right direction. So the arrow points in the direction of increasing fitness. So this kind of fitness graph has many uses. For example, you can ask, where are the fitness maxima? Here you can probably tell it's the one with the green box because it's the one with incoming arrows, right? So there are no fitness increasing mutations that go out of it. You can ask what the adaptive paths are. Basically, you just follow the arrows for the adaptive paths. You can ask, is there epistasis? And this can, epistasis is basically the interaction between genetic effects, right? When the effect of a particular mutation depends on its genetic background. So here, for example, if you look at the wild type, that is the 0, 0, going to 1, 0, that mutation is fitness increasing, but when the same occurs in 0, 1, it's fitness decreasing because the arrow points downwards. The fitness graph approach is very nice for understanding landscapes, but one limitation of all this is that here fitnesses are assumed to be constant, so that by definition kind of rules out frequency-dependent interactions, changing environments, and so on and so forth, right? Which means if you want to do anything with ecology, you can't exactly use this approach. So the problem that I'm trying to resolve is how to develop an analog of the fitness graph for landscapes that are deformable when due to biotic interactions, when you have interaction partners and so on. Right, so and I'm going to sort of demonstrate it to you through a specific example, and the specific example is an evolutionary model of resource competition. And I'm going to split the problem into two parts. Firstly, there is an ecological part. By ecological part, I mean there are no de novo mutations. So we have some C consumer genotypes which are competing for our resources. And the resources are also biological populations. So this is a classic model by MacArthur from the 1970s. And C is less than or equal to R, meaning that's the competitive exclusion principle, which comes out of the MacArthur model, which says that in steady state, you cannot have more consumer types than there are resources. And there are essentially two phenotypes for each consumer, which is the capture rate, not two phenotypes, I'm sorry. There are phenotypes which are the capture rate of resource I by consumer G. So corresponding to each resource, there's a capture rate. And I said two basically because I'm only considering two resources here. So for the evolutionary part, I will now have to specify a genotype-phenotype map. Each genotype has its corresponding capture rate. So that is just a sample I chose for illustration. So now given this ecological part and this fitness landscape part, I still need to specify what the actual evolutionary dynamics is, right? So I'll just do that step by step. Firstly, I'll use, like I said, only two resources for this illustration. And we have to start from a community of consumer genotypes that are in steady state of the MacArthur equations. And we can have only three types of communities, which is one consumer with either one or two resources, or two consumers, two resources. Now, let's say we choose a community as an example, 0, 1. Let's say that is my genotype, and the bracket around it means that it's not just a genotype, but it's actually a community of individuals. There's a certain population size, there's a certain birth rate, death rate, etc. I'm suppressing a lot of those equations here, right? And it's growing on the resources R1 and R2, and the population is in some steady state of growth and death, right? So then step 1 would be to randomly choose a consumer individual from this community and mutate it. So 01, say during a production event, it mutates to, let's say, 00, right? So now what happens? So step 2 is to determine whether this the de novo mutant 00 can actually invade and alter the community. So in this case, 00 can do that. If you compare 00 and 01 on the two different phenotypes, there's a trade-off. So 01 does better, has a taller blue one and a shorter orange one, right? So which means that it can actually invade the community, and this can be worked out by solving the MacArthur model. So if it does so, then you just update the community, and your new community is 01, 00 with their own subpopulation sizes. And step 3, just go back to step 1 and reiterate with the updated community, right? Basically now you have to once again pick either a 01 individual or a 00 individual in proportion to their frequency, mutate it and see where that takes you, and this process continues till you hit a community where there are no more destabilizing mutations, right? And then you have reached the end of this particular evolutionary path. So this is a bit like an adaptive walk, except it's a polymorphic adaptive walk in general. Instead of a sequence of single genotypes, origin fixation, you have a sequence of polymorphic, in general polymorphic communities that are evolving. Right? Okay, so now with this dynamics in place, I can sort of tell you what a generalization of the fitness graph is, and that is a community transition graph. And let me start simply and add them step by step. If we have just one resource, then it's again back to the usual traditional fitness landscapes. Here you have the maxima 01 for resource 1 and 00 for resource 2. You can just check it against those phenotype values. But the interesting stuff happens basically when you add two resources, right? Now you can see that there are multiple kinds of transitions that are possible in the system. So the blue lines give you diversification. For example, if you start at 01 and then there's a mutation like I showed 01 and 00 that leads to a polymorphic community now, right? You have the red arrow, which is a consumer diversity loss. So 1-1 gives rise to a new mutant. In purple over there, 0-1, and which actually outcompetes both 1-0 and 1-1. So now you have, 0-1 comes to displace both of them, right? And there is another further type of transition, and that transition is resource extinction. So let me remind you that the resources themselves are biological populations, so it's possible for them to go extinct. So for example, if you start at the very top, like 1-0, 1-1, with the purple, you have a 1-0 plus 0-0. Now, if you look at 0-0 and the phenotype of 0-0, right, it has a very high capture rate too. That means that it can actually, with that, drive the second resource population extinct. And now you only have R1, and you're back to the old-fashioned fitness graph with no frequency-dependent interactions present. So this is a kind of irreversible transition that leads to sort of diversity loss in the long term. Right? So you have all these various kinds of possibilities. And I should point out again in the green boxes, we have the various evolutionary equilibria in the system. And the evolutionary equilibria are basically, again, they only have incoming arrows, no outgoing arrows. And they are a generalization of the local fitness maxima in fitness landscapes. And those who are familiar with like evolutionary game theory, these are very much like the evolutionary stable states or stable strategies in evolutionary game theory. The difference is that now we have an explicit genetic structure in addition to the phenotypes, which tell us like where these are located, where these stable states are located in the landscape. Okay, so now this was just with two loci. I want to actually show you what a more realistic larger system size looks like, and for that I cannot enumerate all the options. So instead I will have to use a statistical model with L loci, something that has the colorful name of a Ruff-Mount Fuji model. So there are two terms here. First is the additive term with all the genetic loci having a fixed contribution, and then the second term is the epistatic term, which is kind of a noise term which depends on the entire genetic sequence. And in this way, so the epsilon, that coefficient over there, that controls the degree of epistasis. Low epistasis means you have a smooth genotype-phenotype landscape, and high epistasis means you have a landscape with many local fitness maxima in the genotype-phenotype map, right? So with this, one could count, for example, the number of equilibria of various kinds. You can have monomorphic equilibria or polymorphic equilibria. So the red circles, the polymorphic equilibria, vastly outnumber the monomorphic equilibria when the epistasis value is high. And that's because you have now lots of these combinations of this, in a high epistasis, you have lots of combinations of this fitness peaks on the individual resource 1 and resource 2, which can be combined to give rise to sort of polymorphic equilibria which cannot be destabilized, right? So the number is higher for a simple combinatorial reason. It's quadratic in the total number. It's potentially quadratic in the total number of genotypes that can participate in this, right? So from here, you might sort of get the idea that when you have a large system size, meaning a large number of loci are involved, then essentially the number of monomorphic equilibria is a vanishing fraction of the total number of equilibria, so you should always get high diversity, but you should always get polymorphic states as a result of evolution, but that's not exactly the case. It's a little more complicated than that. So to show that, I'm just showing you just a sampled adaptive walk. So what we see above is the number of consumer types and resource types. I start with one consumer type, and I start with two resource types. And quickly, there is a diversification as expected because there are many polymorphic states actually. But then after a while, there is a crash in the population. So you can see that if you look at the lower part, you have the maximum capture rate in the population of either R1 or R2. And near the crash at time 10 or so, the capture rate of resource 1 has gone up very high. And suddenly that drives it extinct. And as a result, sort of now you're evolving only on resource 2. So what happens with the max capture rate in the blue line is that now initially there's a fast drop in it. It is no longer under selection, and there are a lot of deleterious mutations that are not selected against, right? So there's a drop, and then there's a drift, and now you have this black line sort of, which gives you just the selective evolution of the resource of the capture rate R2. So the point is that although there are a lot of polymorphic equilibria in the system, because of this irreversible nature of the extinction of resource, the walks are actually biased a little bit towards monomorphic endpoints. So just to conclude, firstly, I showed you that fitness graphs can be extended to ecological scenarios as well. In this case, it was intraspecific trait variation, but the formalism is more general. And when I apply it specifically to this MacArthur model, higher epistasis leads to greater degree of coexistence, more polymorphic states, but evolutionary paths are biased towards monomorphic populations due to resource extinction. So finally, I would like to thank all the members of the lab of Claudia Bank at University of Bern, and these are the funding agencies. Thank you for your time, and I'm happy to take any questions. Which is comparable to the genetic complexity, then I think for the evolution and diversification will continue for probably a very long time. And also maybe extinction events will be relatively rare. But it's a question of time scales, I guess. Yeah. Yeah.
Yeah, if I understand correctly, this is sort of like an extended adaptive dynamics like model. And so you have like a time scale separation, right, between the
Exactly. Yes, there would be more genetic variation, so that's a drawback of this, but it's also a strength because here you can clearly see that the coexistence or the polymorphism is not driven primarily by mutations, but primarily by the ecological interactions. But yeah, definitely there'll be differences. Yeah.
I've been working on with Yuan Quintam and Shah Moulon at the University of Lausanne. So non-genetic frequency-dependent disruptive selection, or just disruptive selection, can lead to adaptive discrete polymorphisms within a population. And this is because this type of selection gives a fitness advantage for traits that are rare in the population. Disruptive selection can have its roots in different ecological processes, such as sex-specific selection, or social interactions between individuals, or as we're going to focus on this talk today, local competition for resources. So when the trait that is undergoing disruptive selection is polygenic, then this polymorphism faces two extra barriers. So the first one is that recombination and Mendelian segregation of the alleles are going to make it hard for the two discrete morphs to emerge. And then once that they have emerged, hybridization between the two morphs is going to continuously produce intermediate phenotypes that are maladapted. So in order to study disruptive selection of polygenic traits, there are two kinds of models. So the first kind of model comes from a population genetic approach, where they're looking at the genetic association between alleles at different loci, and they typically consider the case of local adaptation. And here they find that selection on modifiers to suppress recombination between co-adaptive alleles is usually favored. This kind of suppression could be thought of as inversions, for instance. And there's a second school that would use models where what evolves is the effect size of an allele on the phenotype. So this type of model usually considers local competition, where the two morphs arise due to character displacement. And here what they find is that usually the genetic basis of this polymorphism remains into a single large effect locus. So in this project, we're broadly interested if the nature of disruptive selection, if it's due to local competition or to local adaptation, if it influences the evolution of the genetic architecture. So today, because I don't have that much time for this talk, I'm going to focus on a more specific question, that is, do recombination and effect size modifiers respond similarly to disruptive selection? At the end of the talk, I'm going to give kind of three other things we're doing. So in order to answer this question, let me introduce you to our eco-evolutionary model. So here we're thinking of a population that is subdivided into patches and that can consume two types of resources. So here I'm showing them as purple and blue, for instance. So in the case of local adaptation, I'm going to consider that each patch either has the blue resource or the purple resource. Whereas in the case of local competition, I'm going to consider that each patch contains both the blue and the purple resource. So then the life cycle starts by adults exploring the resources in the patch that they live in. And this competition is mediated by a trait Z that gives you the preference, the resource preference of an individual. So here I'm showing you in this horizontal axis the resource trait, where you can find the resource purple and the resource blue. And you can see one bell-shaped curve that tells you the attack rate of an individual on those resources. So in the example I'm giving you here, this individual would attack the resource blue, but not the resource purple. And there is a niche breadth that can control how much resource around the preference of an individual it can consume. Then once this individual has consumed resources, there is local mating, where the fecundity of individuals is proportional to the amount of resources that they have acquired. This offspring that they produce is going to disperse among all patches uniformly and compete for breeding spots in the next generation. So all the ecology is mediated by the trait Z, so let's give this trait a genetic weight. So I'm going to consider that the trait Z is encoded by 20 genes. Each gene is composed of 3 elements. So the first element is a binary locus, a plus or minus allele. So you can see this as an adaptation to consuming purple or an adaptation to consuming the blue resource. An effect size modifier that tells you how big this adaptation is, and a recombination modifier. So the genotype to phenotype map is just summing across all 20 genes the maternally and paternally inherited copies of the binary locus that I'm representing by A, so it's +1 or -1, times the effect size modifier that I'm representing by gamma. And I sum across all 20 genes of an individual to have this individual's phenotype. Then there's a mutation dynamics on these elements. So the binary locus mutates from plus to minus and minus to plus at a given small rate, while the modifiers, they follow the continuum of allele model, and they mutate by small step mutations. Then, during the production of gametes of an individual, there is the recombination that is given by a recombination rate, where the recombination between two adjacent loci is just given by the mean between the paternally and maternally inherited recombination modifiers at that position. And what I'm going to show you here today is based on individual-based simulations that I performed on SLIM. So let's start with the results. So the first result I want to share with you, I'm considering that all these 20 genes are freely recombining in the chromosome. that so there is no mutations in the recombination modifiers. This recombination is fixed and free, and what can evolve is the effect size modifier. So what I'm showing you here on this slide is for the case of local competition, I'm showing you how much the genetic variance explained by one locus changes in time, so across time. So each line you see is one locus. So you can see that all 19 loci that I'm representing by the black lines, they all have a genetic variance that is going to 0, meaning that you're fixing the gammas, the effect size modifier, and the binary alleles in these genes, while one gene that I'm showing you here in green is explaining all the genetic variants of this trait, of this consumption trait. So this leads to a concentrated genetic architecture on this one locus. And we do not observe that in local adaptation when dispersal is low. So here you can see that in the same time scale, you still see many different loci contributing to the phenotype. And when we increase the case of local adaptation, you increase the dispersal between the patches, you kind of recover a little bit the concentration, but not into one single, but rather a few genes. So a few genes are controlling this phenotype. In fact, if you wait long enough, you're going to see the concentration. So if I could summarize these results into 2 sentences, I would say that disruptive selection leads to a concentrated architecture into a single segregating gene, but this concentration occurs much more rapidly in local competition compared to local adaptation. So for the second result that I want to share, it's kind of the opposite. So now I'm going to let recombination modifier to evolve while keeping the effect size fixed. And I chose an effect size that in order to have an adaptation to consuming one resource or the other, you need the whole 20 genes to be either plus 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 or minus 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 across all 20 genes. So here I'm showing you in the case of local competition the mean recombination rate between pairs of genes, so from 1 to 20. And in lighter colors you see free recombining alleles, while in darker colors you're going to see recombination suppression. And in fact, if we let this simulation evolve, quickly we see recombination suppression. So for instance, in less than 10,000 generations, you can see that suppressed recombination between gene 1 all the way to gene 10. And if you wait a little bit longer, you're going to see that step by step, this recombination suppression region is going to increase until the recombination is suppressed throughout the whole chromosome. And then this leads to 2 haplotypes in the population, and this is as good as this population can do. It stays there. And we do not see the formation of haplotypes in the case of local adaptation. We do see a reduction in recombination, so you can see that there are some darker colors, but it's not the formation of the haplotypes that are so clearly seen in the case of local competition. So disruptive selection again is leading genetic architecture to concentrate on 2 segregating haplotypes, but again this concentration is much more rapidly and completely in the local competition than in the local adaptation, because local adaptation selection is already keeping the good alleles in the good patches. So as a last point, I wanted to show you the difference in the signatures in the phenotype that we observed between these 2 cases. So here I'm showing you plots as a function of time, where each dot is an individual, and I'm showing you the phenotype of this individual. So individuals that have the phenotype 2 can consume the blue resource, individuals that have the phenotype -2 consume the purple resource, for instance. And then you can see that in the case of local adaptation, you have 2 types of individuals, so 2 morphs. One morph is consuming the blue resource, one morph is consuming the purple resource, and from time to time you produce, via dispersal, hybrids between these 2 morphs that are badly adapted. Whereas in the case of local competition, you can see that you have 3 morphs. So one morph that is the homozygote consuming the blue resource, and then you have another morph that is heterozygote and is consuming the purple resource, while the other homozygote is badly adapted, is not adapted. So the difference between local adaptation and local competition in this case is that local competition you have a well-adapted heterozygote, while you do not see a well-adapted heterozygote in local adaptation. So with that, I would like to give you some take-home messages that summarizes my results. So concentrated architecture is favored by disruptive selection in both cases of local competition and local adaptation. But in local competition, selection on genetic architecture is much stronger because you produce more hybrids because the two morphs coexist locally. So it occurs in different time scales. Local competition is much faster. And you observe different signatures. In the case of local competition, you have an adapted heterozygote, while in the case of local adaptation, you have adapted homozygote. Some ongoing work, we're studying the evolution of dominance, where we show that dominance only evolves in the case of local competition and not in the case of local adaptation. We show how inversions grow in these two models, in these two cases, by strata of different size. So local competition, the inversions grow by small strata, while in local adaptation, the inversions grow by large strata. And we're showing that because in the case of local competition, the homozygote is badly adapted, it causes a lot of genetic drift in these individuals, and they can accumulate recessive deleterious mutations. So with that, I would thank you for your attention and let you know that U1 is attending the conference in Montreal, so feel free to reach in. So thank you for your attention.